So what we're talking about today is pedigree diagrams. And these have been around a very long time, longer than DNA sequencing. And they've probably been doing this before the advent of an understanding what DNA is. And essentially what this is a family history. Okay, circles represent females and squares represent males. And of course they have offspring. So in this case, I have a female and a male having two children, a daughter and a son. This son didn't marry or have a couple and have children, but this daughter did marry someone who was outside the family. You can see that. And had 5 children. And in these 5 children, I have these solid black colored in squares. So this, of course, they had a couple of sons. They had 2 sons and 3 daughters, and what we can see is that 1 son and 1 daughter out of the 5 children from this mating, okay, are infected by an autosomal recessive disorder. Okay, now before I get into that, I want to speak to about what pedigree diagrams have been used for. So before, again, DNA sequencing, before we could actually take someone's blood and sequence out the code that we've been learning in class and find the genes that we know, because we've mapped out the human genome, we know exactly where all the genes of the human body are. So we're able to take DNA from blood samples and actually map out what disorders people actually have. So and we've done that successfully. So if you want to know if you're going to have an inheritable type of recessive, in this case autosomal recessive disease, you can do it by DNA testing or DNA fingerprinting, sometimes they call that. However, before you could do that, we were using these diagrams. And what these diagrams are is a family history. And in this family history, we look at phenotypes. Now, in the case of recessive diseases, and many diseases are recessive. Let's step back for a second. We know that there's 2 chromosomes. On the chromosomes that are homologous, we have one from the maternal mother and from the fraternal. And on the locus here somewhere, we have a similar gene. And this gene might be dominant that came from your mother, but it might be recessive that came from your father. Now, the recessive gene that we've talked about is that broken gene, okay? There's some kind of what? Some kind of mutation. Maybe one of the nucleotide pairs has been mismatched and now it codes for the wrong amino acid that makes the wrong protein, because the protein's now going to fold based on different amino or R groups, is everything we've learned, and have a different shape and a different function. And this if you're copying code from this allele and it's random which one we use in cells, okay, we will make the ineffective protein that doesn't make something. In the case of, let's say, sickle cell anemia, which is a recessive disorder, we do not make the hemoglobin protein that helps us deliver oxygen in our red blood cells correctly. In fact, having one wrong nucleotide pair leads to an amino acid that's different, that leads to a different shaped protein that makes the hemoglobin a different shape, so that instead of these circular kind of donut shells that are flexible, you get these cyclic type of cells that can cause blockage and deliver oxygen poorly, that we've talked about in sickle cell anemia. So to have full-blown sickle cell anemia, like all recessive disorders, you have to be what? Homozygous recessive. You have to have that phenotype. Now, in Hardy-Weinberg, we've talked about that the phenotype that we know, the genotype always, is what? Q squared, and that's the homozygous recessive individuals. What we do not know, okay, if you're what? Heterozygous, or you're homozygous, okay? These guys give the same phenotype. So that's the issue and the tricky part. So in a recessive disorder, because it's about the mutated gene, okay, wrong code for the wrong amino acids for the wrong protein, okay, the only thing that we're going to see in our family history is if you have the disease or you do not. Okay, so why do we have them and how are they useful? Well, again, before the advent of DNA sequencing, a couple may know that they have a family history of a disorder in their lineage, in their family, and they know it's passed down. You don't need to know what DNA is or if DNA in fact is the heritable compound to do these types of diagrams. So if you know that heart disease or some kind of, let's say, sickle cell anemia runs in your family, then of course, okay, you could try to use a pedigree diagram to try to predict what the chances that either yourself has it if you're this individual, or what your offspring will have. Now, in a case of sickle cell anemia or any other recessive disorder, some of these could be deadly, and sometimes people want to choose and family plan according to their family history. I've known couples that decided not to have children because they know there was a great chance of them having offspring with very deadly diseases. It's a very, very touchy and very, very personal type of information. And so these pedigree diagrams helped potential families or couples, make decisions based upon should they have kids or not. Now, it also may be that, hey, when am I going to have the disease? Now, sickle cell anemia is certainly a disease that is immediate, even from child, but there are some diseases that develop over a time period, and you may, and so some people would want to know if themselves are going to be inflicted based upon the family history. Okay, so this is what we're doing. We're going to take the family history based upon identifying the phenotype of the recessive, in this case, because we're talking about autosomal recessive, and try to work backwards to figure out information about the rest of these individuals. Again, very accurate way of determining the genotype based upon the phenotype being expressed of the recessive allele. But I will say, okay, it's not perfect because we can only really work backwards, meaning I have some questions possibly about the grandfather pair. Remember, this might be grandmother, grandfather, and this is the mother, the father, and this is the grandkids, okay? So we really need to be able to work back, and we have to have accurate information about siblings and, let's say, possibly deceased members of your family that you do not know if they had a disease. So if you do have a good family history of who had this heritable disease, you can get some very accurate information and be able to, what, from this couple, predict, okay, what the percentage or possibility of your children having that disorder. Okay, and again, people family plan based upon pedigree diagrams for a long period of time. Now, DNA sequencing kind of has replaced pedigree diagrams, but I will say, oftentimes we can't DNA sequence people that have passed away, okay, or you may not, or the people, let's say the siblings, the sibling from this, the sister and brother, maybe this sibling doesn't want to be DNA tested. So this type of work with pedigree diagrams using family histories to look at the chances of yourself developing a disease or your children possibly is usually the work of genetic counselors, and they'll either build this through looking at phenotypes or again using some DNA sequencing. And it does, I just have to say, it's very sensitive, personal, and sometimes very heart-wrenching decisions have to be made or people make based on this information. So we're getting into a very personal area. Although we're doing these pedigree diagrams, understand their use of them is through many families that causes a lot of pain and personal angst. But in any case, moving forward to give you that, we're going to talk about basically 5 different types of diagrams that show different types of hereditary and how these types of diseases, okay, can be passed down, and show you the limitations and show you the amount of accuracy you can actually come up with based upon these. So we're going to start with autosomal recessive, and I'm going to clean up this area here, and so that we can work with it. The first thing you have to realize is that autosomal means chromosomes or chromosomes that, or information on chromosomes that is not sex, okay, related. So we're talking about information on chromosomes that do not have any sex linked abilities, meaning the gender of the individual does not help determine the disease. These diseases are not carried on the X or Y chromosome, they're carried on the autosomal chromosomes, the non-sex linked. Now, very important, now when you talk about a non-sex linked, and this is really important as you look through these, non-sex linked, okay, um, disorders, what you're going to see is the amount of males and females, okay, that can have these diseases is proportionate. So that's one way to see that. So this is autosomal non-sex linked. I see that there is a inflicted individuals that are approximately males and females can have that, okay, and we'll talk about the chances. Now if I scroll down to sex-linked genes, you'll see that in this case, this X-linked recessive gene, you see that there's a proportionate, or in this case, you have only the what? The males that are inflicted here. So that's one way to look at these right away. I'm dealing with autosomal recessive. I know that, boy, I tell you, the amount of males and females that can have these are both equally can happen, or the probability is the same 50-50 because it's got nothing to do with the X or Y chromosome. Now, one thing you realize when you're dealing with a recessive disorder, we're dealing with, of course, that broken gene, and to show the phenotype, okay, to show the disease, whether it be hemophilia or PKU, okay, we need to find the individuals that are Q squared, or just, you know, hey, that are showing, if they're showing the expressing this disorder, they must be homozygous recessive. So that's what's speaking here. But something very important, in a recessive, okay, type of inheritance patterns, and that's what we're trying to build here through the family history, what we're going to see is that we're able to skip generations. Okay, and then you'll see the appearance here, and this is kind of like we did test crosses in Mendelian genetics. You can be able to skip generations. So anytime you see or want to recognize a recessive pattern through family history, it's going to be able to skip. And the reason why it skips is, if you probably haven't figured it out, we have something called carriers. Now carriers, I'm going to use big A for dominant, little a for recessive. The carriers, okay, as we've talked about, they carry the broken allele but don't express it because they have the other chromosome, okay, from the other other pair, that's producing enough of the good protein to mask, okay, the bad protein that we're making here that doesn't do a certain function that leads to the disease, okay? So there must be carriers, okay? So the biggest thing when a recessive is that I see that there's a skipping of generation, meaning there must be carriers that are bringing that alleles together, okay? Now if I scroll down, all right, if I go to autosomal dominant, I don't see a skipping. And if I go to an X-linked recessive, now of course this is due to the genes being on one of the, well, in this case, by saying X-linked, we're saying on the female X chromosome. We see mostly males here. We'll talk more about this when we get to this one, but you still see what? A skipping of the generation. So this recessive type of inheritance pattern is due to making of those heterozygotes, which are the carriers. So back to this one again. So what can we do with this? All right, so what's really helpful when you're dealing with these pedigrees diagrams to figure out, you know, the chance of, or what people might be, okay, in terms of their genotype with the known phenotypes only, because we're not really dealing with DNA, is what we do is we're going to do some crosses. So what do we know? Well, we do know that we have some children here that are, okay, showing the recessive allele. So what we have here is the cross. Now we don't know the phenotypes of these guys. Well, we do, I'm sorry, we do know the phenotypes. They're not expressing the disorder because they're not filled in, but we don't know their genotypes. We're trying to build the genotypes here with a very basic, what, dominant and recessive type of thinking in this genetics. So we're using Mendelian genetics here, which we can because a lot of recessive alleles have a broken gene, okay, with broken proteins, as we've talked about. So how do I go after this? Well, okay, I look and see where my expressed individuals are. A boy and girl are both being expressed here in this third generation. Now, working backwards, what kind of cross gives me this? Well, first and foremost, if I do a cross or do a Punnett square here, I know that the father and the mother of these individuals are not showing the what? are not showing or expressing the disease, whatever that recessive disease may be. So I'll put the dad on top, and so what's going to give me? Well, if I was to say, well, they're both homozygous dominant, you could probably quickly see there's no way that I could have homozygous recessive individuals. If I do all of this, you can see clearly that there's no possible way. So this, knowing the phenotypes, which are now recessive, okay, knowing the phenotype and of course now the genotype, little a recessive, I know that the only possible combination this must be, or at least a good possible combination, would have to be that they are carriers. Okay, so when I do this again, I got little a for, let's say, the father. We'll put the fraternal on top, and then I've got the mother with the same carriers. They're not expressing it because they have one of the dominants. I'm going to make big A, homozygous dominant. I'm going to make another carrier. I'm going to make another carrier, and lo and behold, I make that. So really, the only combination I have is that these both must be heterozygotes, and that's how you can find out, going backwards, okay, what the parents must have been. Now you say, Mr. Gradsky, oh, what if I was to put an A here? Okay, a big A. What if one of the parents was, because they're not showing them. Well, if you do your Punnett square, okay, what are you going to get? You're going to get no individuals that are homozygous recessive, yet we show that. So from the what? If I was trying to figure out, if I was a child here, and I was trying to figure out the genotypes of my parents, knowing my what? Knowing my sister and brother's affliction to the disease or not, their phenotypes, I'm able to narrow down the possibility, or now at least understand that the genotypes of my parents must be heterozygous for both. Okay? Now, I do know that if I was the daughter of this parent, so remember, I'm a grandchild, okay, in this case, a granddaughter of this family. If I work backwards, I know that I have an uncle, okay, so remember uncle here, and he's not inflicted, but I don't know what their phenotype is, okay, but it's important to know that my parents had this. But if I wanted to work backwards, okay, maybe he wanted to have some information, and he knew from the children, he didn't have any children, but he knew from the children of his sister and another individual, those were the outcomes. Maybe he can work back to see what were the possibilities of his parents, or in this case, the grandparents. So what are the possibilities? Well, we know that we're producing little a. What do we do know? There's no infliction of the disease, so none of these individuals are homozygous recessive. So what are the possibilities? And again, now we're getting limited here. Well, one of the possibilities is certainly that we had two carriers again. Now you say, okay, little a, big A. So if we had two carriers, all the possibilities would be this again, and we could make someone who's inflicted. Yet we didn't see anybody in this generation. You may say, well, Mr. Gradsky, I don't see anyone inflicted, so this can't be. But wait a minute. Remember, it's all chance. Only having two children, maybe the third child, just by chance, hook or crook, would have had that double A. So there is a possibility, okay, we don't know this one, there is a possibility that both parents could be carriers. What about the possibility that one of the parents was homozygous dominant and the other one? We have to have at least one of them be a carrier. Okay, so what's the possibility that one of the parents was homozygous dominant? Well, I would say that's another possibility. And you say, Mr. Gradsky, I don't like this because now you're just giving me possibilities. The only thing we're able to really come up with certainty in this pedigree diagram is that both mother and father here had to be heterozygotes. But here, okay, if I had big A, little a for, let's say, the father, okay, and then I had big A and big A homozygous dominant, you can see the types of crosses we would have here. And you can see clearly why the sister had a good chance of being what? A carrier. And possibly, if this son of this group or this uncle of this was thinking about marrying and having children, okay, maybe he is double A. He's got a chance, but we don't know for sure. So this pedigree diagram, although it's given me some definite information about one couple, okay, we need more information. Okay, so what we would need a what? We need a previous generation here to help me. So I'm getting as much as I can from this pedigree diagram right here. So if this son of this grandfather, grandmother, granddaughter, grandfather of this child, or the mother and father of this individual, the uncle of this, we can't really speak to a huge amount of confidence exactly what it is, but it's either going to be one of these two. And not knowing any information previous, we're stuck with, yeah, you could be either a carrier or you could be homozygous dominant. Of course, they do not want to be a carrier, because once you have a carrier like this group, there's a good chance if the other one's a carrier to express the allele. So what we would tell this individual is that there is a possibility that you are a carrier. Okay, and unfortunately, that's as much information we were able to tell these individuals in genetic counseling years ago. Now they can actually sequence the gene of this individual and find exactly if they have that recessive allele or not. Okay, so that's what this tells us. Let's go move on to autosomal dominant. Okay, there are types of diseases where you only need one dominant allele. One classic case is something called Huntington's disease. It doesn't get enough publication. Huntington's disease, or HD, is a really, really impactful disease that not that no disease impacts them, but this one is a fatal disease that causes the loss of mental function and the physical function. If you know anything about Alzheimer's, you kind of lose your ability to think clearly, memories, and who you are eventually become a shell of yourself. And if you have Parkinson's, you tend to lose your motor control. You'll be able to move. Huntington's a combination of both, and it winds up being fatal. They wind up dying from this, and this is something you do not want to have a family history of. Unfortunately, it is there, and it's an example of an autosomal dominant. Autosomal meaning it's a gene on a chromosome, and that chromosome is not on a X or a Y. And what's different about this one is, unlike the recessive disorder, because you're making carriers here and not expressing an allele, the next generation could happen. All right, but here you notice every generation, okay, is expressing this allele. And what's different about it is that you only have to have one dominant allele. Okay, what this means is that the pair of chromosomes, again, an example of and we have a locus here, and we'll do the maternal and the fraternal, and one of these that's dominant, okay, is basically a gene, probably from a mutation. So it's kind of a weird thing, but it's this protein is made that's wrong, but that wrong protein, okay, starts a set of sequences or side reactions that starts in play the whole loss of memory and motor function. So it's such a only have to have one being made that this impactful protein. So remember, most recessive broken alleles don't do anything, and the resulting disease is because you don't have the protein to do something. This mutant form actually sets in motion a bunch of side reactions that causes the disease. So this is an autosomal dominant. All you need to have is one of them. So if you're expressing an autosomal dominant, the first thing I recognize is that I'm going to have it in every generation, because in order to show this, I'm either what? A or little a. This is not showing it, so this must be what? Recessive. So what I do know from this pedigree diagram that I didn't know in the other one is the individuals that are not expressing, let's say, Huntington's disease are going to be homozygous recessive. And the ones that do express the disease, they're either going to be what? They're going to be either homozygous dominant, or they're going to be the heterozygotes. Just having one A produces that really toxic protein that catalyzes all those different reactions that leads to loss of memory and motor function. So we do have some information here, and certainly individuals in a family history here want to know, what are their chances? This individual wants to have kids with another mate, and it wants to know what's the chances of it having the allele, okay, or broadcast an allele. Now, you may say, well, of course you don't have it, but in the case of Huntington's disease, this disease starts kicking in, mostly for late 40s to 60s. So you could be living a very normal life, and then all of a sudden you start to have show the symptoms of Huntington's disease. So you are going to usually couple and mate and have children in a family way before your 40s. So you don't know if you have it. And so, oh gosh, what do I do here? Again, this is a very sensitive and personal type of issue here. So what are the chances of this individual? Well, let's look what we have here. Look at the family history. And of course, it's easy to recognize this as being dominant. Notice it's not a sex-linked gene. It's not an X and a Y. There are just as many females, okay, as males have the disorder. So if we look at the first cross to get a feeling of this, we had an individual, a male, okay, who did not have the one of the big A alleles, and we have the female who did. And now we do not know if it's what? Big A, little a, but we kind of do, because look at the outcomes here. Let's think with you, and this is where pedigree diagrams become important here. Look at our outcomes. If in fact my male was homozygous recessive, then you can see what's going to happen. Every offspring is going to be a heterozygote, and that means every offspring is going to have a dominant A, and therefore every one of these boxes should have been blackened out to show that every single individual here is going to express the Huntington's disease. That did not occur. So therefore it cannot be, okay, big A, big A. It must be that this individual only had one of the bad alleles, okay? So let's get rid of this part here. So this individual should be big A, little a. Now if you don't see that, do the cross. You get an individual who's going to have Huntington's disease because it's a dominant disorder. You're going to have another one, but here, because the other individual has to be homozygous recessive, you're going to have some individuals that do not have the A. And that's what this pedigree diagram is doing. Okay, so this has to be little a, okay, because for the same reasoning, we have to have some individuals that are not inflicted. So we know definitely all of these phenotypes, okay? And so now we can make some guesses what these individuals are. And of course, what are the chances of this individual expressing, well, it doesn't have any of the genes. This person, okay, if you notice, what are the chances? The chances are 50% if you have in this case a father who has Huntington's, okay, that's a heterozygote, and you have a mother who's homozygous recessive, okay, of course it has to be, the mother is not inflicted, you've got a 50-50 chance of having that. And of course this is here. So this individual, okay, genetic counseling wise, okay, if he didn't know he had the disease, without knowing anything else, he would know and live his life thinking 50% chance. He had a 50% chance of having what his father's going to die of, or what his other siblings will die of in their 40s to 50s to 60s. And that, again, is heart-wrenching. Okay, when you do these, understand where these things come from and the importance. Okay, now, it just so happens this box isn't filled in, so he is going to wind up being the individual who doesn't have it, but until they get genetically tested, they do not know. So the pedigree diagram is telling us that he doesn't have it, but he doesn't know yet. He has to live his full life. So do I get genetically tested and find out, or don't I? Because if I find out I'm going to be like my brothers and sisters, do I live my life differently? These are all, important personal questions. Okay, but you can clearly see from our analysis, our logic, we're able to find, okay, the phenotype. So from all this work, I've got to the point where, okay, these individuals, all of these family members had a 50-50% chance. Now that doesn't mean when you look at these diagrams, you're going to see 50% show the infliction, because remember, this is all chance right here. There is a chance if you roll the dice, low numbers, that all of these could be. Okay, remember, if you roll the dice 1, 2, 3, 4, 5 times, is there a chance of having all heads? Yeah. So you just can't look at it that way. All right, but that's important you see that much so far. Okay, so in number 3, I'm showing you a family history of an X-linked recessive disorder. Now, these disorders are sex-linked genes, meaning the broken alleles, the recessive disorder or the broken gene is on the X chromosome. Okay, and this is important. This is where we first learn this using the Morgan Drosophila experiments that we talked about in your last homework. Okay, so these are X-linked genes, meaning the male and female alleles or characteristics that we get by being a male or a female. Okay, the recessive disease, okay, is attached to that. A great example of a sex-linked gene or an X-linked is hemophilia. And hemophilia, okay, has been really been done very much with the history or the family history of the noble family of England, because hemophilia is a disease where individuals have a tough time forming blood clots. The protein factors that help blood clotting aren't made correctly, so these people can certainly bleed out and lose their blood by very simple cuts. Okay, so it's definitely a phenotype that is recessive, but it's that recessive or that group of information or that gene is on an X chromosome. So if you've got an X chromosome, and if you're a female, okay, now females have 2 X chromosomes. So they can have the recessive disorder and with that broken gene for that protein it helps in blood clotting. And then they can have the dominant, the working one. Now males, okay, now males are XYs, they have 1 X. So their Y chromosome doesn't have the clotting factor. They only have 1 X chromosome. If that is recessive, and that's important, if that is recessive, they don't have another X chromosome, as we learned from our homework, to mask that A. And so this guy is going to do what? Result in showing that phenotype. That's very important. Now, of course, if it gets an X that is dominant, it doesn't. But because a male only needs 1 X chromosome that's recessive to show the disorder, it's going to happen. There are going to be more males who have it. Now, for a female to show an X-linked recessive disorder, it needs to have 2 X's that are both recessive. It does occur, but it's less frequent because you need to have both of them, and the chances of having both recessive alleles is a lot smaller than just having one. So males are expressed. So this is a recessive disorder. I taught you in the autosomal recessive that we can skip what? Generations. Why? Because we have carriers that can help carry the broken allele, okay, to the next generation. But to know it's sex-linked by looking at these diagrams, you'll see a larger portion amount of males having these disorders than females. It doesn't mean that females can't happen, but they're going to be more rare. Okay, so that's how I look, and that's how I know this. The first thing I look at this and say, hey, is this an X-linked recessive? Is this a autosomal dominant or a autosomal recessive type of inheritance pattern? I see only the males really are distributing, and I'm missing a generation, so I know it's X-linked and it's recessive. But let's dig into this a little bit. So the first thing that jumps out of us is that the males are inflicted or expressing the phenotype, which is recessive. We know that by skipping a generation. We know it's an X and a Y, and the Y chromosome doesn't have it, but the X does. So we'll draw the recessive here. Same thing for this, okay, XY, and it'll be the recessive here. It doesn't have another X to mask it, as we talked about. Now let's try to find out more information about the rest of these individuals. They may want to know what their genotypes may be just based upon the phenotypes of others that we know about. Now we know the rest of these individuals are not inflicted, and so the males that are not inflicted, we definitely know that these guys, guess what, are X and Y again. And of course, they have the big A. This is again another X with a big A. They have the dominant allele and the Y. So all of these males here, X to the big A and a Y. It's the females we have questions about, because the females okay, could be carriers. Now we know that females, okay, could be carriers, and if they could be shaded in, it's rare that you have females that are homozygous recessive, but we again have to look backwards and look at each other. So let's get into this. So we have a male, X to the big A to the Y, and then we have a female. We don't know what the possibilities are, but let's just make two suggestions. It either could be what? X homozygous dominant or X heterozygous. Those are the two scenarios. So let's do both. Let's do X to the big A, the homozygous dominant, and let's see what we get here in these crosses. Okay, so we're going to get, okay, an X with a large A, and then an X with a small a, and then we're going to get an X with the big A, and another Y, here's the male, and then this was a big A, so this is X to the big A, and then the X to the little a, and then we have the what? The male, which is going to get the X from the big A, and then of course the Y. And often you'll hear is that the sex of the offspring is determined by the male, because the male can either give an X, the female only gives an X. So if the male gives the X chromosome, it becomes a girl. If the male gives the Y chromosome, because it only has the Y, only boys do, it becomes a boy. So you'll hear that the male or the father determines the sex by what chromosome it delivers in the sperm to the other gamete. Any case, in this cross, what do we get? Well, we get any inflicted individuals? None. Okay, we got no inflicted individuals in this cross. We do, though, have some carriers. We have two female carriers. So based on the results of this, this second generation, it looks to be that there's a good chance that this is the phenotype, because the children are not expressing. More importantly, the males are not expressing it. All right, so probably a great chance that this is the phenotype, but we're not 100% certain about this. Let's do the other one. Let's do if we have heterozygote for the female or the mother here. So here, and then let's do the recessive, and then we still have the father, who is going to be again only has one recessive allele, so it's going to express it. And so when we put these together, we get X to the big A, X to the little a, and then the other females, we're going to have some females that could be what? Inflicted with the disease. Okay, we don't have that here. Okay, and then we have male that's not going to be inflicted, but then we're going to have a what? A male that is. So we don't have males that are. So it looks to be that this is the best guess scenario, but we're not 100% sure. Isn't there a possibility with the two rolls of the dice that I rolled a male, okay, that's not inflicted? So the answer is that we're not 100% sure based on this information what exactly the phenotype of this grandmother of this family tree, okay? But I guess we can be pretty certain, though, that the male here is XY, okay, and we can produce them what? In both crosses, and we are producing females that are not what? Inflicted in both crosses. So we can't say, based on this information, exactly what the phenotype. It's either this one or this one. However, what must be the phenotype here? Well, if you notice, we're skipping a generation indicative of the recessive type of inheritance pattern, but we're seeing allele pop up here. Okay, now if I know that the male is not infected, remember this female is the daughter of this grouping, and this male is not showing this disease. This disease could be hemophilia, we talked about, and so this does not carry any of the bad alleles here for that disease. So if we have some offspring that are expressing the recessive allele, it must come from this guy. So based on this information, there's no question mark, this female must be X big A, little a, must, and that we definitely know, because this one's not bringing the broken little a allele. It's coming from this. We're expressing it, so it's coming from the mother. Okay, that's why they call it an X-linked allele. It's coming from the mother. The father cannot bring this because the father does not make what? Okay, the father in this case, because it's not inflicted, okay, is not giving a Y that's inflicted, and he only has an A that's not inflicted. So it must be the allele is given here. So we definitely know. So from the pedigree diagram, we can figure out the mom is a carrier, okay? And that's all we know from this, but you should be able to recognize this. And, you know, another X-linked recessive allele is colorblindness. Now, it's not a disease, but colorblindness is more it happens more so with the males, and it follows the same patterns because the colorblind gene, or the proteins that help us perceive colors from our eyes, okay, are located on the X chromosome. All right, let's move on. Okay, so number 4, okay, we're looking at another inheritance pattern, another pedigree diagram. This one's a little bit different. We're not skipping a generation, and if you notice, there's a lot of males and females both having this. Okay, so what's going on here? Well, let's take a look here. This is an example, okay, of an X-linked dominant. Okay, I'll write it here, X-linked dominant. Okay, inheritance pattern or pedigree chart. Now it's a little different than the autosomal dominant, okay, and let's see why. Okay, first of all, if I look at my male here, it's an X and a Y, and we know something about the Y. If I'm not expressing the dominant allele, I have the recessive allele. Okay, so the female is expressing the allele. Now it's double X. Now it either could be double A, or it could be what? X, big A, little a. Okay, again, we don't know which one yet. Let's see what the second generation tells me. Okay, well, party people, I have a female that is not inflicted here. So this must be a female that is X little a, if this is an X-linked dominant gene, allele. So what's going to give me that little a? Not this guy. If we cross the male X with the broken allele, or at least the recessive, I should say, in this case, and we have the Y, okay, if it's the dominant allele is the bad one here, okay, and then I have the female. If it's double A dominant, you can see that every female is going to at least have one dominant allele, correct? And if that's the case, we can't possibly make a daughter from this cross to not be inflicted if just having one dominant allele makes the disorder. So this cannot be, based on this evidence here, okay, this mother, okay, cannot be homozygous dominant. It's this guy. So we just found that there. Now if you don't see that, again, put in a recessive here, and you'll see that little a will give me the homozygous recessive, which is not being inflicted. It's safe, because in this case you want to be homozygous recessive, because if you have the X that's dominant, it's shown. All right, notice the males, and a lot of the males and a lot of the females will be inflected. So if we do this cross, okay, where we have X to the A, okay, and we did the half of it already, so we're using this recessive now. This is going to be X little a y, and then of course X big A y. What can we see? We can see that having one dominant, okay, allele ensures us that 50% of the boys and 50% of the girls will have the disorder. And you know, we are approximately seeing that between the two girls, one had it and one didn't. Now, the percentages won't always be perfect in small roles, but boy, that really is supporting this finding here. But clearly this individual has to be homozygous recessive if having one X-linked gene that's dominant does that. Okay, now if you notice the boys and girls are inflicted a lot here, and that's again indicative of the X-linked dominant allele, or inheritance pattern. And of course these boys would have to be what? X to the A, And then Y must have that non-dominant A. And of course, these girls must have what? Well, they must be, if they're inflicted, they must have at least one dominant. We don't know, okay, about the other. Or do we? Or do we? Okay? And what information does that have? Maybe this girl wants to know if she's homozygous recessive or dominant based upon who she's going to marry and have kids with to give the percentages of the next generation. So do we know? Well, look at this male. This male is inflicted. Okay, if it's inflicted, what must it be? Yeah, you're getting on this now. Good. It's got to be an X with a dominant A with a Y. So it has to be this, because if this male is inflected, it's only X must be the what carrying that bad allele that makes some kind of protein overtake the entire disease. So we know we have a male X to the A, Y, and then we have, in this case, X to the little a, X to the little a. And what does our cross give us? It gives us a carrier. And we're just looking at percentages now, because maybe they just need to know what their percentages are, and that will help them. X to the A, Y. X to the little a, X to the dominant, that's the bad one's the expression, and then X to the little a and Y. So if you notice, party people, in this cross, okay, that we only have, we have the female. If you notice, all of the females, really important, all of the females made will be what? XA little a. So now from doing that cross of these two, okay, clearly the females have to be X big A to little a. And just by using expressions of the phenotypes, we're able to get genotypes and build a family history. Of course, it's based upon information that's solid. And again, this is a very non-common one, okay? And it's not about just having a dominant allele, it's about how the sex will influence the dominant allele. Unlike the autosomal dominant, which is just having a dominant allele, this one is definitely influenced by the X and Y. Notice again, okay, this male was delivering, okay, the bad allele. And it's because the male determines a sex, all of the what? All of the females became ill here. So that this generation really lets you know that there's a sex linked here, because all the females were inflicted in this cross. And why? Well, because the male, which was delivering the X for females, was the bad allele. Okay, and so that's really what makes this a little bit different from the autosomal one. All right, so moving on to 5 and the last one. I know this was long, but hey, I am tall. All right, now we've got this cross. So what's so different about this pedigree is that all the males are expressing the disease here. Okay, there's no exceptions here. This is an example of a Y-linked, okay, inheritance pattern. And you say, well, why? Well, because it is. The Y has, in this case, to be a boy, you have the X chromosome and the Y, and of course on the Y chromosome you have the male characteristics, although the genes that make us a male and make us different. But on that there's also a bunch of genes for male characteristics, and I think a lot of Y-linked disorders have male characteristic problems being expressed. Maybe not producing as much testosterone and all those things. Okay, maybe possibly being infertile, but the point is that Y-linked disorders are due to a problem of a gene. It gets a smaller chromosome, so there's not as many genes here, but some problem with the gene on the Y. So to express it, you have to have the Y that's inflicted. So this male is XY, and as we learn, just having this Y in order to mate, okay, to make a male, you have to deliver the Y. The male makes the sex in the children. So the X came from, all right, the mother. If it's a male, the Y had to come from, and again, that Y is inflicted. There's a problem with the gene on that Y that's causing a problem. You want to call it recessive, you can, but there's only one Y, so just having the Y creates the disorder. Okay, so all of these are getting the Y chromosomes from the father, and of course these boys are getting their Y chromosomes from this father, so it's essentially a Y-linked trait disorder, okay? And that's really, really easy to identify. And that's as far as I'm going to go there. In any case, please use these descriptions in helping you figure out all the other questions. Keep in mind, the biggest ones are recessive disorders that skip generations, dominant disorders, and of course, sex-linked, where the X and Y plays a role in the expression. Okay,